Thanks for viewing Day 2's materials. Today we're going to be talking about proteins. Cells produce tens of thousands of distinct proteins, and they're really the taskmasters of cells, and they're required to carry out essential functions for the cells, including catalysis, making up structural proteins, helping cells move, and much more that we'll talk about in today's material. So in the first two videos, we're going to be talking about amino acids. Those are the monomers that make up proteins. And in video 3, we're going to talk about how proteins fold and how this helps them execute their unique functions in cells. So let's dive into amino acids. Amino acids are the monomers that make up proteins. So proteins are the first macromolecule that we're going to be talking about in Bio 161. And as a macromolecule, this means that they're large molecules made up of smaller subunits. And we call those smaller subunits monomers, meaning one part. And the monomers link together or polymerize to form polymers or many parts. And that's shown down here with a monomer joining this growing polymer. Of the macromolecules, lipids are the only macromolecule that do not form a polymer, but we'll talk more about that later. So let's dive into what an amino acid looks like and how it forms polymers. So most proteins are made up of just 20 amino acids. And amino acids are composed of a central carbon that's bonded to the following: a hydrogen atom, an amino group, and remember the N and the N in the amino group, this functional group. The central carbon's also bonded to a carboxyl group. Here's the OH in the oxy. And an R group. And the R group, or R, stands for this variable side chain that we'll talk more about in video 2. Okay, so let's take a look at what this amino acid looks like. So I'm going to start by showing you the basic structure of the amino acid. So we have our amino group and we have our carboxyl group. So with the carboxyl group, that carbon is double bonded to one oxygen and then also has a single bond to another oxygen. And these are joined through covalent bonds to a central carbon. And that central carbon is bound to a hydrogen and then a variable R group. And so this R group changes depending on the amino acid that we're looking at. But in a cell, because a cell is mostly made up of water, amino acids look like this here. So this is called the ionized form of an amino acid. And as we talked about in the lecture on functional groups, this amino group behaves like a base. And so it attracts protons from solution. And so this nitrogen has a positive charge. And then this carboxyl group, this oxygen behaves as an acid and it donates a proton to solution. And so this oxygen carries a negative charge. And so this is how most amino acids look like in the cell. Okay, so this is our basic monomer. Next, let's talk about how these monomers join together to form a polymer. So I want to define some key terms. So the first term I'm going to talk about is a condensation reaction. And when I'm talking about a condensation reaction, there are two things to remember. The monomer goes in and the water comes out. So kind of think about a condensation on a glass, right? That condensation is coming out of your glass or out of your cup and kind of making beads on the glass. So here's a picture of a condensation reaction where we have our growing polymer and a monomer goes in. And so this monomer attaches to the growing polymer and water comes out. Now we're going to talk more about how this process occurs inside cells, but I want to point out here that polymerization usually requires energy, and that's because entropy or disorder of the system decreases. And so we'll talk about the laws of thermodynamics, but I do just want to point out that this is an energy demanding process to polymerize a polymer. The opposite reaction is hydrolysis. And in this reaction, water goes in So hydrolyze, hydro meaning water added, hydration, and the monomer comes out. And this breaks apart polymers by adding a water molecule. So here we have a water molecule. It's going to go in a chemical reaction, and then water goes away and the monomer comes out. So again, hydrolysis is the opposite. So it's usually can happen spontaneously and releases energy because entropy or disorder of the system increases. And we'll talk more about this next lecture. But again, just be thinking about how when you build something together, that requires energy. And when you break it apart, that can happen spontaneously. It doesn't require an input of energy into the system. And the system in this case is the cell. Okay, so let's look at this for an amino acid. So let's look at how a condensation reaction happens and how this results in a bond between two amino acids. And we call this bond a peptide bond. So here are two forms of our ionized amino acids. Okay, and I'm going to draw your attention to this oxygen here, and then these two hydrogens that are joined to this amino group. Okay, so remember, this is a condensation reaction. So a monomer comes in and water comes out. Okay, so these bonds will be broken. Ta-da! And these atoms will go ahead and form new bonds to create a water molecule. And that's shown down here. So now we have water, and these will come together, and the bond that forms, this is a covalent bond that forms between the carbon of one amino acid and the nitrogen of another, and this is called a peptide bond. The other thing I want to point out here is that because we've lost this oxygen, and it went away to form water, this has turned from a carboxyl group that was bound to the OH to a carbonyl group. Okay, so when you're looking for peptide bonds, you can kind of look out for these carbonyl groups because they usually occur right after that. Okay, so as the polymer grows, we have different terms to use to describe those polymers for a peptide or for a protein. So if a chain has fewer than 50 amino acids, it's going to be called a oligopeptide, which means few peptides. So less than 50 means few peptides, and sometimes this is just shortened to peptide. A chain of more than 50 amino acids is usually referred to as a polypeptide or many peptides. And then the term protein refers to the complete functional form of the molecule. So we're going to be looking at in class some examples of oligopeptides, so just really short sequences of amino acids. And then once they're fully folded, we'll call those proteins. Okay. Sometimes protein is used to describe any chain of amino acid residues too. So if you have a question about what we're referring to, definitely feel free to ask, and I'm happy to clarify. Okay, so in class, we're going to look at some polypeptides or oligopeptides, oligo meaning few. And you should be able to count up how many amino acids are in this oligopeptide. And so if you use my way, so if you count after these carbonyl groups, we have 1 amino acid, 2, 3, 4, 5. And that means that after each one of these is a peptide bond. So if we have 5 amino acids, that means we have 1, 2, 3, 4 peptide bonds. And we'll practice this more. I have a question here for you to pause and look at and try to see if you can identify the R group, right? So if we were looking at this, right, we have our central carbon here, right? And this is now bound to it's a carbonyl group, and we have our amino group. We should have our hydrogen, right? And then this would actually be our R group. And so there's actually an amino acid where the R group is just another hydrogen. So here we have our R group. So be prepared to kind of analyze these structures. And you should also be able to see that when we make these, we call this a peptide bonded backbone. When we make these structures, you can see that there's there's a difference at the start of the polymer and at the end. So at the start, we have an amino group, and sometimes we call this the N-terminus. And we have a carboxyl group at the end, and we call this the C-terminus for the carbon that ends. And so this gives us some nice features for this peptide bonded backbone. We have that directionality that we just talked about. The way in which amino acids join together also orients the R groups in a particular fashion. So they're all facing the same way. And so when they're in water, they're going to interact and fold based on their chemical properties. And so this is going to be really important for their function. And around this central carbon, these bonds here, the atoms can kind of rotate and be flexible. And so this gives the protein some flexibility to move and form conformations that are energetically favorable. So these are just some features of this backbone. I'll also mention, because we're talking about it right now, that we can number these amino acids. And so starting at the beginning, so starting at the N-terminus or the amino functional group, we have the first amino acid. And so the amino acid, when it's just hydrogen as the R group, this is called glycine. And we would number this 1. And also we have this 3-letter code here. You don't have to memorize this at all for this class. That it represents glycine. The next amino acid here is a proline, right? And this would be number 2. And then here we have a serine, 3, 4, 5, and so on. So this is how we would number them. And so here are the amino acids that are joined together by this peptide bond. So in summary, amino acids have an amino group, a central carbon, a carboxyl group, and a R side chain. And if you're following along, you'll probably notice I'm missing. They should also have an extra hydrogen, so we can add that in if you're taking notes. Peptide bonds also are what join together amino acids in a condensation reaction, so water is lost. And the peptide bonds in an oligopeptide create directionality, they orient the R groups, and they give a protein a flexible backbone. So that's it for this video, and in the next video we're going to be talking a lot more about these R groups.